So thank you, Brett, for that kind introduction and for not making people have to stare at me in two places at the same time. It was very kind of you. And thank you for having me here today. I'm very excited to have an excuse to come visit your beautiful country. I've enjoyed it quite a bit. I'm a little disappointed that clip didn't work because now I can't remember what it said, and it was a good segue into what I was going to be talking about. But one of the main reasons I'm excited to be here today is I do a lot of business presentations, I do a lot of science presentations. But I got into this field for one reason, and that was to improve patient outcomes. I was frustrated as an academic doing research and seeing the disconnect between not the desire, because you don't get into science to not want to help people. It's not lucrative enough to do it for any other reason. But to survive as a scientist, the incentive structure is not necessarily conducive to helping people. It's when Brett showed that chart about how the information transfer goes, scientists to journals to doctors to patients. That bottleneck at the journal side of things is a really important thing that I want to emphasize before I move further. Your career is judged based on how many papers you publish. And as a result of that, the emphasis is always on publish, 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 publish. Your work is supposed to be to a pretty figure that goes in a paper that makes people ooh and ah, and it's not meant to disseminate to you guys. And there's not a lot of incentive for scientists on the academic side to do anything different. At least not in the States. I know some other places have more of a private interest in funding research, but in the States you have a group of scientists that decide who gets money. So this is really cool for me. I enjoy interacting with patients. And because of that, there's a lot of cool things that I want to talk about. And so forgive me if I bounce around a little bit, because I want to tell you about my cool research also. But there's some other pet things that are important to me that I hold dear to my heart as far as patient advocacy that I'll talk about as well. So first of all, what's a Novoron? So Novoron is a company that I founded. It was a spin out of my research that I did at the University of California, San Diego. And we began as a group originally working on spinal cord injury. And technology has evolved into some MS therapies as well. We're going to talk some more about that. We're also going to do a little quick overview on MS. I'm sure that you're not the group that I need to educate on that too much. But then I'm going to talk about ROA. And for those of you who aren't familiar with the role of ROA in MS, I'm going to emphasize that a bit. Then I'm going to talk about what I think is the primary kind of missing link in MS treatment right now, and that's healing lesions, repairing damage, reversing some of the deficits. And then I'll talk about my little pet stuff, the MS advocacy stuff. And when I do that, hopefully I can not get too in depth, because Brett and I both wanted to take the chance as we do the joint kind of panel discussion second, leave some things to spark some discussion. So Novoron, we originally were founded in January of 2013. We were originally licensed from the university, two pieces of technology that are kind of the foundation for our company. We've been completely privately funded. We've raised about $1.5 million through the National Institute of Health, and that's funded all of our research so far. We've been fortunate enough to incubate in the Johnson & Johnson incubator in San Diego, which has been tremendous because they have so many more resources than we could have. But that's all boring stuff. This is the part I want to talk about, because this is what's going to be the kind of the founding principle for everything I talk about going forward. What Novoron decided we wanted to be about when we started, that was different than a lot of other places, is we decided we were going to develop our treatments to the best possible place to avoid any chance of false negatives. And what I mean is not every trial fails because the drug doesn't work. Not every study fails because the intervention isn't real. Sometimes it's poor study design, and sometimes people take shortcuts with how they design their drugs, how they design their trials. And we decided that despite the fact that there's shortcuts you can take to get to value inflection points, to get more grants, to get a paper, to get somebody interested in throwing money at you, that wasn't worth potentially sacrificing at the end of the road whether or not patients benefited. And so I'm going to talk about not only how we're going about that, but how some of the ways that I want you to think about this as well, and how you assess research, and how you hold people accountable for the research that's being done to benefit you. So this is our founding team, and I really have no purpose for showing you this, except for I wanted to add this. This is a picture of me at the Waitomo Caves, which was one of the coolest things I've ever done, and so that's up there now. We can skip this though. So all of you are familiar with what multiple sclerosis is. Autoimmune condition, it attacks myelin. Myelin is the really important stuff that makes your nerves work the way they're supposed to. And when myelin's not there, they don't work, and eventually the nerves die. You can't get them back, permanent dysfunction, all bad. Nobody wants that. Here's some examples of anesthesias. Here's some chewed up myelin. I really like this picture. It looks like moths came in and eat this. I like that. But what I want to emphasize here is MS research has done a great job of developing disease-modifying agents. We've done a really tremendous job at reducing the number and severity attacks that patients have. And here's a little chart of all the places where we've done that. So here's your blood, here's the brain, and here's all the lymphocytes and the T cells, and here's the drugs that block them from doing all the bad stuff that causes attacks. Here's the problem. This is all great stuff. We've done nothing here. Once that damage occurs, we do nothing to repair. This is the missing link in how we cure this disease. If we stop the disease from progressing, that's fantastic. But for those of you that are here, it doesn't get you back to where you want to be. It doesn't restore the function that you have lost. And as a result of that, this is what I think is the next leap that we need to make in MS. So, how do we do that? Well, here's the thing, and some of you might know this, but I have the feeling a lot of you don't. Your body remyelinates all the time. If you get a concussion, you get a bump on the head, if you get a cold, a lot of times you have these focal demyelinating states. Your body does a great job of fixing that. And here's the process. I kind of want to emphasize a little bit of this. So here's the normal white matter of your brain. Here's the normal healthy myelinated axons, and here's an MS lesion. Or we'll pretend for the sake of argument it's any lesion where there's been a demyelinating event. Here's your progenitor oligodendrocyte. So the precursor cell that makes myelin and coats nerves. These cells will infiltrate into the damaged area, and then they'll divide. They'll just make more soldiers to do their job. Once they divide, they'll mature. And as a mature oligodendrocyte, now they can go back and repair the lost myelin. In multiple sclerosis, this doesn't happen. But it doesn't happen because it's blocked here. It's specifically blocked at this step. You still get progenitor cells that infiltrate your lesion. They still divide. They just don't mature. And this process, and this is what I really want to emphasize, this process is mediated by a molecule called Rho. And Rho hyperactivity in oligodendrocyte progenitors is what shuts down their maturation. As a result, let's make Rho not activate and we can make these cells mature. Like this. Fix it. So here's the problem. Rho's not unique to MS as far as a therapeutic target. So there's a lot of companies that are working on drugs that target Rho. The problem is, Rho's not a bad guy. It's not a mutation, it's not a pathogen, it's a normal molecule in your cells doing its job like anybody else that's important for cytoskeletal remodeling, focal adhesion, migration, motility, division, all sorts of things that cells have to do all the time to work correctly. Unfortunately, not all, but a good majority of the approaches that are being developed by large pharma right now target Rho directly, either via interfering RNA or small molecules that just shut it down. They're called pan-Rho inhibitors. And even though that can be great for shutting down all the bad stuff over here that we don't want, you're also getting some effects over here that you don't want, and that's what causes toxicity. And that's what causes negative effects that limit the amount of drug that you can use to get the effect you want, so because you need to balance all the bad stuff that's happening with the good. So Novoron said, okay, well, we know that this is a very unique system over here. So what I want to emphasize, these events that happen are receptor specific. So Rho is a very active molecule, it does a lot of things, but what it does is really responsive to who tells it to do what it's supposed to do. So, you might be a really good employee, but if the boss that's in charge of PR tells you to do PR, you're going to do that. You're not going to run off and do accounting. And the same thing is happening here. When Rho is activated through this very specific sub-signaling pathway, that's when things go haywire. So we decided we need to target this. We want to focus on this pathologic signaling pathway so that we can leave the normal functions intact, which means we can use more drug, get better effects, and reduce the potential side effects. So here's a little breakdown, blowing this up and looking at it more specifically. We know this pathway pretty well. We know what's causing this Rho activation. But the problem is, these little guys here that are responsible for leading to the Rho activation, there's like 9 things that can flop in and out. So targeting any one of them doesn't necessarily recapitulate a beneficial effect, because other guys can come in and take over. So you knock out P75 and his good buddy Troy comes in. Literally his name is Troy, it's a protein called Troy, and Troy takes over for P75 when he's not hanging out anymore. So what do? And this is where Novoron's technology comes in. What we discovered is that this protein called LRP1, the cell surface protein, which is really valuable towards, it's almost like a chaperone on the cell surface. When things get bound or stuck or signal, LRP comes over, sticks to them, takes them to where they need to be, and then to turn them off, sucks them into the cell where they get broken down. Otherwise, things on the cell surface that get turned on never really get turned off. The only way you can turn them off is to bring them inside. Well, it turns out that while everybody was looking at all these really important things that are binding and interacting, the trafficking of this protein complex, the movements that actually led to this signaling event, were being characterized but not looked at very closely. And it turns out that those events, the necessary events, nothing happens. You can get this whole thing forming, all this stuff coming into combination, but without the movement, no signal happens. And so that's LRP1. And if you knock out LRP1, so basically we took oligodendrocyte progenitors, neurons, made them so they can't express this protein, and we treated them with molecules that would normally cause this Rho activity. And when LRP is not there, you can't turn on the Rho. And so that was exciting. So what does that tell us? Yes, this is a central component. But there's other components involved that are also central. The difference is this is necessary. There's no replacement. Nothing else can recapitulate the effect. So now we have a single target that can actually be a master modulator of this cell. So we developed a library of compounds, of which our lead compound is now Novo117, which acts to block this interaction. And there's a couple reasons this is cool. Not only do we block this specific Rho pathway, but we do it on the cell surface. We don't even have to figure out fancy ways to get into this cell. We act outside, so that's a whole barrier of entry that we don't have to mess with. But by blocking this interaction, we, in response to numerous molecules, and these are just 3 examples, in response to numerous molecules, our drug blocks that Rho signaling. Now, as I mentioned before, Rho is important for a number of diseases, one of which is spinal cord injury, which is where we started. And so while we're talking about encouraging remyelination and maturation of oligodendrocyte progenitors, we're also talking about nerve growth. And that's easier to look at. It's the only reason I'm telling you this, because I have pretty pictures to show you, and I want you to understand what you're looking at. So here's a nerve in a normal situation, happy, growing, doing its job. But if you take that same nerve and you put it in an environment of what you would see in a damaged spinal cord or in an MS lesion, not so happy. They're stunted, they ball up, they sometimes die, but they can't extend neurons, okay? They can't regenerate. Ready for this? Magic drug. Oh, look, they grow again. Yay! That's the type of regeneration that we're looking at, okay? So, how do we look at this in MS? Obviously, now keep in mind, the remyelination and the nerve regeneration are tremendously linked in both spinal cord and MS, and other diseases that we're looking at as well. But I uncouple them for the purpose of this lecture because remyelination is the conceptual no missing link. So I'm going to focus on the remyelination, not the nerve growth, beyond the pretty picture that I showed you. So we already know the system. We already know what's shutting it down. Novo117 is supposed to reverse this. So how do we test this? Well, I'm going to show you this picture. And this picture goes back to not Novo117, which is the cool stuff, but because, like I said before, we're all about minimizing false negatives. So before we do the rigorous development of the drug, we said, let's make sure that this target, LRP1, is still important in MS. We already showed it was for spinal cord and for nerves. Let's make sure it is for MS. So we created animal models that only in those oligodendrocyte progenitor cells, so not in every cell, you can't take LRP1 away from all the cells in an animal, it never gets born. But only in those oligodendrocyte progenitor cells, and we induced demyelination. And for those of you who are interested, we used 3 different models, 2 different demyelination models, lysolecithin, cuprosome, and EAE. For those of you following along at home, if you know what all 3 of those are, brownie points to you. The moral of the story is, in all of our models, we saw tremendous benefit when LRP was not present in those cells. Basically what you're looking at here is green is myelin proteins. Okay? Here's a normal healthy tissue, they're hanging out doing their thing. Here's after a demyelinating event, in this case the lysolecithin, there's a lot less green. Here's 5 days after they've been healing for a while. The cells are mounting a bit of a remyelination attempt versus a lot of a remyelination attempt. And here's the numbers that back that up. So you see a significant increase in the myelin protein production when LRP is not deficient. So that's our target validation. Now, what I'm not showing you is our drug data. Because just because we validated a target doesn't mean that we can just throw a drug in an animal and it's going to magically work the same. That's where the development part comes in. And I'm going to really hammer on this development part more later, so I'll stay away from it for a little bit right now. But what do we know? So we know that LRP1 is a viable and important therapeutic target in MS. We know that loss of LRP1 restores myelin and neural regeneration, and that Novo117 is our lead compound that's capable of doing this. And then the rest of this business knowledge. So, I'm going to talk more about the development, but I want to talk more about you guys now too. So, Novoron really only exists for one reason, and that's because the whole system of drug development has changed. It used to be that universities and academics would find cool research, they'd identify a new target or a new way to treat a disease, and Big Pharma would come in and say, good job, we're going to make a drug, hit that target, we're all off to the races. There was no in between, that's how it worked. As companies grew bigger and became more successful, there got to be, I like to call it, too many cooks in the kitchen. They become public companies with shareholders and board members, and the willingness to take risks on a large number of targets got lower and lower and lower. In addition, our diseases became more and more difficult. We got away from monogenic diseases and very simple diseases of like scurvy, where you just take vitamin C and you're all set. That's a bad example, but it's a really simple example. So what happened is you got companies that became risk averse. Costs went up, the tolerance for risk went down, and so really innovative stuff stopped being pharma's forte. And that's when you started seeing this phenomenon of spin-outs and startups, where both universities and big pharma were spitting out little companies, little satellite companies, to do riskier work left and right. So basically, big pharma sitting back and saying, all right, 30 companies, you go do the crazy stuff, we'll wait till one or two of you do it well, and then we'll buy you back and we'll develop it from there. Which is great. There's a couple reasons why that's great. Having high risk tolerance in smaller groups of people allowed you to do the crazy things that bigger groups were never going to agree on doing. Okay? The bad news is it also took a tremendous amount of resources away, both in personnel and in capital. So, harder problems, less resources. The only thing you got going for you is the innovation from the people that were willing to take on these tasks. So, the barriers have changed. It used to be that money was not the problem. And now it's about how are we picking winners and losers? How do we facilitate the success of those riskier companies? And how do we assess them in this new kind of paradigm of drug development? So understanding this concept and understanding how this model has changed, I wanted to plant the seed so that you can think about how you want to start viewing your research and how the things that you want to view as valuable and the things you want to put your weight behind as far as an advocate. And the three things that I think are really important here, and why I think what Brett's doing is so important, is you need to be informed more than anything else. Here's the thing. Public companies, in companies like mine, we require value inflection points. And one of the things I hate about going from science to business is all the buzz cliche words. It drives me nuts. But value inflection point is one that you hear a lot. How do you get to a point where you say, we're more valuable now because we accomplished X? We took a drug and now we made it IV deliverable. We know its composition. We saw these effects. We tested in this animal model. Every one of those things is a value inflection point. But for a company like mine, we're never going to do a clinical trial. We'll have to at some point take on a partner, a Pfizer, a Merck, a Johnson & Johnson, somebody who has the big guns, the regulatory lawyers, to partner with us to develop. And so our customer is always big pharma. So we're constantly trying to put together data packages that adds value that makes them look at us favorably. The problem is that gets really risky because just like media wants to tell you exciting things so you read papers, we want to tell pharmaceutical companies crazy things so that they want to work with us and lend us their resources. And that gets to be a slippery trap because you can paint a pretty picture without doing all the rigorous science that you need to be able to do to really give them something to work with that works well. Because you have limited resources, a lot of people will say, what can I do with $6? How far can I get? Okay, that's all I'm going to do. I'm going to use that, and if that's not good enough, I'm moving on to the next thing. So when I say Novoron really committed ourselves to not doing that, that's why we haven't taken any private money. Because private money says, great, let's throw it into people. Well, we haven't developed it yet. We haven't done the rigorous things that need to be done to make this the best drug, not just a drug. Okay? So when Brett's talking to you about good scientific communication, what he's really trying to tell you is Everybody in the world has different interests. Pharma, business people, stock markets. They all have different reasons to look at these pieces of information, these pieces of news. And really, you're not really one of them. Your interests are your own. So, informing yourself, knowing the truth, reading between the lines, that's an important thing to be able to do, and picking your sources as well. The main reason that I'm here is because when the more I got to know Brett, the more we started having conversations, the more I realized that he was trying to do something the right way, that was also the harder way, and I could relate to that. That was something that I thought was very a valiant idea for him to have. Once you are informed, the MS community has no problems being motivated. Being informed, maintaining your activity in these groups, combining your efforts, combining your voice, and speaking up for the things that you need are hugely important, and being consistent and pushing for change that you need. But none of that matters if you're advocating for the wrong things, or if you're catching on to the latest trend or cliche. And that's why we talk about, what Brett mentioned, like with stem cells. That people try to purposely make stem cells this umbrella term because people still assign value to that. And I can tell you personally, the vast majority of stem cell approaches since the late 70s, early 80s have failed miserably. Bone marrow transplants have an actual scientific rationale for why they might work. The tests have not been good. We still have a lot of work to be done there, but it is a personal choice. The risk reward, where are you now? How much chances that might be true, and is that worth it to you? But a lot of these other stem cell therapies that people are traveling to Mexico and Guadalajara for, that just are putting a huge stem cells, fly here, we're gonna put stem cells into your veins. There's no scientific justification for that, but people, they link the word stem cells together. And that's, sorry, a little bit of rant, apologize me for it. So here's another question for you. And I'm genuinely asking, if I told you right now that once a week, I'd stab you in the eye with a needle, but you would be cured. No more attacks, no more disability. How many people raise your hand if you would do it? Raise your hand if you would not. So about 50-50? 50-50 is a pretty good market on a disease that's about a 1 in 1,000 prevalence. But the people that didn't raise your hand, why? Somebody raised who wouldn't do it. Why would you not? I did not hear you say it again. Well, so assume that it worked. Assume that we're talking about a long-term clinical trial. We know it works. If you get stabbed in the eye, no doubt about it, you're cured. Once a week, though. Would you change your mind? Would you do it now? That's a valid point. Now you're getting into some crazy stuff over there. Appreciate that, though. Let's pretend we're in a public health system. So honestly, if it's a cure, governments are going to jump all over that. They will make it available because MS is a really difficult drug to treat long term. So, although once a week, let's pretend it's free. Let's pretend the only cost is stabbed in the eye, just for a moment. Would anybody change their vote?
But you can still see out of that eye.
Oh no, yeah. Only cost is the weirdness, and it probably wouldn't even hurt. Let's pretend it hurts though. Let's just pretend it hurts. No disability with the eye though. It's just the weirdness of getting stabbed in the eye. Is there anybody who now knowing all that wouldn't do it? Okay, then I was terrible at explaining it the first time. I apologize. The reason I bring this up is because this is an actual conversation I had with a pharmaceutical company. We were talking about some Alzheimer's therapies. They approached us about whether or not we thought our approach to neuroregeneration might work in Alzheimer's. And my response was, we have a lot of development left to do. If we were to do it now, we would have to do direct administration into the brain. And he said, we're out. And I was like, okay, that's fine, but why? And he's like, because we would never do that. Not because it wouldn't work, but because they don't want to take on a project that requires something that sounds hard. Literally, that was his answer. It doesn't matter if it worked. If it cured Alzheimer's, we still wouldn't want it. And I was floored. By the way, this is Johnson & Johnson, one of, I think, the biggest pharmaceutical company in the world. They wouldn't want it.
Wouldn't make them any money.
Money's not the issue. It's the PR. So this is a huge conglomerate public entity. They have boards, they have shareholders, and they have to convince all sorts of non-scientists and non-physicians that, hey, we're going to start stabbing people in the eye. In this case, it wasn't an eye, it was drilling holes in the skull. Equally scary. Didn't want anything to do with it.
Do people like Johnson & Johnson have the final word
Yes.
Well, what about the hospitals and the doctors in the hospitals? Don't they have a fund?
So I would love to get more into this later, but I'll answer you briefly now. The answer is yes, they do have a fund. I say that being an American. And in our country, we're very capitalistic. Other places that have a strong, large population where the hospital systems are united, they might have enough lobbying support to be able to push for things. But at the end of the day, hospitals can't develop drugs. And hospitals can't convince people to take on projects. And hospitals cannot tell you, we guarantee you profits. And that's how the system works. And don't get me wrong, I'm not demonizing the profitability, because here's the problem. In academia, our incentive structure is publications. They don't even necessarily have to be right. You just have to trick people into thinking it is for a little while, and you get a paper out of it, and you get a promotion. There's no reverse validation of what you've done. It's a valiant effort. It's a very altruistic concept, but there's nobody that's checking your work and making sure you're right. It's just kind of this hodgepodge of information that we're meant to sift through and say, what's real, what's not, and how can we benefit patients? On the pharma side, there's a very real check and balance to this. As opposed to having this plethora of data where we have to question whether or not what's good and what's bad and what's real, what's not. Pharma will not pursue something unless they are positive. Because of the financial incentives, pharma's kind of motto is fail early, which sucks if they quit because they think it's hard, but it's great because their only thing is, does it work? Brett can attest to you, a lot of researchers, you have a PI, a postdoc, an undergrad, a grad student, and at some point, you as a worker, your career is built on if you publish. And if you have a hypothesis, and it turns out the hypothesis is wrong, it's really hard to get that published. It's really hard to say, hey, you know what's really cool? I found something that doesn't happen. The problem is it should be really cool. The hypothesis validation shouldn't be a necessary component of academia, but the system needs to change. That's another thing I'd love to talk about more. But having profit incentives does drive pharma to things that work, so they're not trying to trick us or push stuff down our throats that we don't need. So it can lead to some greed. It does have some negative effects, but we can talk more about that. Let's see if I'm not getting into this. So talk a little bit about this. Big pharma will make sacrifices based on marketability, speed to market, things like that, but that's why companies like us exist, and hopefully we're trying to overcome some of that. I already said that. I got ahead of myself there. So Novoron's mantra, avoiding false negatives. We're all about development, and because of that, one of the hardest things for me is when we did the interview with Brett, or when we've had other press releases. Lately, Novoron's actually had a lot of good press in the States. People start contacting me, and I try really hard to respond to all the patients that contact me, asking me when they can participate in our trials. And I have to tell them when we're ready. Because I am not going to put a product into clinical trial just because it's good enough. Because I would feel terrible if something failed just because I took a shortcut. Because Pfizer was willing to buy me out because I got to this level, and I said, great, I'll take that check. I'm going to come back to New Zealand and hang out in caves for a little while. That's what we don't want to do. As a result of that, we are holding on to our technology longer than what tons of people tell us to, so that we optimize and make sure it's the best possible product that we have before we partner with people, because I don't think that it's in their scheme, in their best interest, to take an asset and then spend another 5 years developing it themselves. Maybe they would, and that would be fantastic. Not a risk I want to take, because once we get to that point, you can't go back. We can talk more about that later, because I think I also didn't explain that very well. So things that you need to think of when you're informing yourself. When you read these studies, if you don't know what model it was, if you don't know how it was administered, if you don't know what species it was, and if you don't know what any of those things even are as far as options, those are the things you have to know in order for you to be an informed advocate for your disease. Okay? And people like Brett are the people that can help you understand those things. And Brett loves doing those things, which is weird, but he does. But do not. He left out this thing from the John Oliver show. Al Roker on the Today Show, at the end of that clip was like, he said, I think the key to scientific studies is just finding the one that works for you and then believing that one. He literally said that on national TV in a scientific segment, and like nobody questioned it. And I threw my TV out the window at that point. I said, this is ridiculous. Because that is not what we should do, any of us. So, that's pretty much, I've covered all of it. I was just going to talk about some of the very specific things that we worry about when we're developing our drugs, things that you don't necessarily think about. There's the very standard things versus the things that you don't think about, like stability, handling. How can you freeze and thaw? What type of fluid should you store it in? Crazy stuff that a lot of people don't think about. And what I do want to leave you with is one kind of thought is, I do drugs. Protein-based drugs was a little weird, but still drugs. And that process is linear, and it's been established, and there's regulatory agencies know how to handle them. Cell therapy is different, because it's a cell. A cell is composed of thousands and thousands of proteins, lipids, DNA, all sorts of things. It behaves the way it wants. It moves, it divides, it grows, it shrinks, it does with all these crazy things. And as a result, the rigidity of how we optimize cell therapy is a much more difficult process. And as a result, people aren't really trying that much. There was a study that was done in spinal cord injury. In rats, they injected stem cells into a spinal cord, saw some benefits, went to humans. They injected 2 million cells into a rat spinal cord. Rats are tiny, spinal cords are tiny. And they said, well, 2 million worked in a rat, let's do 2 million in humans. They did it. They spent millions of dollars to do 2 million cells into humans. And guess what? Oh, it didn't work. Really? That's crazy. You injected 2 million cells into something that's this big as opposed to so small that I can't even demonstrate it to you up here. And those are the types of things that cell therapy is just now catching up on, is how do we rigorously define a development process for cell-based therapies. Not just bone marrow transplants, but stem cells, precursor cells, things like the mesenchymal stem cells where they secrete things like that. So that's that. I'll end with this. Another picture of me, Brett, and I in the caves. Thank you for your time. I apologize I went over time. And we'll take a break now, I guess. And then we'll come back and you guys can ask some questions.